I'll start by briefly covering direct RNA sequencing, which is a technique where we can generate many results from a single experiment. Shown here is a transcript isoform from its 5' start site down to its 3' poly A tail end. And this is what direct RNA reads should theoretically contain. From these reads, we get multiple data types. So we get the splicing pattern, which allows us to accurately quantify different isoforms. We can find any novel exons or connections, identify RNA modifications, as well as quantifying the poly A tail length. And so without direct RNA sequencing, we would have to perform a series of different experiments to obtain all of this data, which is both time and resource intensive. Now we get all this information from a single experiment, and so this gives us many avenues to investigate. Today, I'll mainly focus on those from isoform quantification and also the RNA modification M6A. The brain has complex mechanisms which regulate its gene expression. Two of these are RNA splicing and RNA modification. Alternative splicing allows for the production of multiple isoforms from a single gene, and this greatly increases the diversity of proteins that can be generated. RNA modifications affect many cellular processes, such as splicing, as well as RNA translation efficiency and degradation. The most abundant RNA modification is M6A, and the human brain has very high levels of both splicing and M6A. While M6A has been relatively well studied with short-read sequencing, it's difficult to obtain a single molecule resolution. So typically we get just a peak on a gene body of the M6A distribution, and this doesn't often give us the exact position of the modification or the which transcript the modification is actually present within. Now with direct RNA sequencing, we get both of these, so we can get the exact position as well as transcript single molecule resolution. Our aim was to apply direct RNA sequencing to 3 brain regions to investigate how different gene regulatory mechanisms contribute to overall brain function. So we used post-mortem brain samples from 4 control individuals, and these came from 3 different brain regions. So we had the prefrontal cortex, caudate nucleus, and the cerebellum. These were all sequenced on the PromethION device. So because direct RNA sequencing allows us to accurately quantify isoforms, we were able to identify widespread changes in isoform expression between the brain regions. We can see in the PCA plot on the left here that the samples are clustered by their brain region based on their isoform expression. On the right, the plot shows us where all of our differentially expressed isoforms are coming from. So we can see most of them are specifically upregulated in cerebellum, followed by those that are upregulated in both PFC and caudate nucleus. So to illustrate an example of a brain region-specific isoform upregulation, I'll use the SYNPR gene in our data. So we found 4 isoforms for this gene, and overall gene expression was highest in caudate nucleus. We can see in the isoform expression plot on the right that only one isoform D, is contributing to the total gene expression profile in the cerebellum. Whereas for the other brain region, we get expression of all the isoforms, and it's spread out a little bit more evenly across them. This isoform D also had a different upstream protein coding exon, and so a slightly different protein domain. Moving on now to our modification results. We observed a typical enrichment of M6A around the stop codon and in 3' UTRs. This was also consistent across all of our different brain regions. We found that approximately 50% of our detected isoforms were modified with M6A, and that the cerebellum had the highest proportion of both modified genes and transcripts. We also found that the cerebellum had the highest ratio of modified reads per M6A modification site. We were able to identify both common and specific modification of genes and isoforms between the brain regions. So as we can see in these plots in the dark grey bars here, if a gene or isoform is modified in one brain region, it's typically shared across all the brain regions. However, we do also see a specific modification of genes and isoforms, and most of these are in the cerebellum. We took the genes from this analysis and used them in a gene ontology analysis. We found that the commonly modified genes were showing association with microtubule polymerization, protein modification, and synapse organization. Those modified specifically in either caudate nucleus or the PFC showed more neuronal-like terms and neuronal signaling terms, whereas the cerebellum had the most distinct profile here with terms more related to DNA binding. I also just wanted to highlight with these red bars here that approximately 40% of the genes that are specifically modified in a brain region have upregulated expression in that brain region. And so brain region-specific modification isn't simply due to upregulation of expression in a particular region. So as well as identifying both common modification and specific modification, we can also identify changes in the levels of M6A at a particular site in a transcript isoform. And we can find when these are actually changing in their levels across different brain regions. So in total, we found approximately 1,000 genes and 1,500 isoforms with differences in the M6A modification rates. In the plot on the right here, we can see that most of the time, these are coming from the cerebellum, more than 70% of the time. And this makes sense because we had the highest proportion of modified genes and isoforms here. I've got two examples from two different genes. The top one here is NEAT1, which is a long non-coding RNA. This actually had a decrease in the M6A modification levels, so that's shown in the red on the top here. And so that goes against what we would typically expect from the cerebellum. On the bottom, we have the SNAP91 gene, and this shows the more typical increase in M6A levels in the cerebellum. Going back to our expression analysis, we can see that even if we subset genes to only those related to M6A modification, we can see that our samples are still clustering by their brain region here. Shown in red, we can see cerebellum has the highest expression of M6A writers and readers, and these are the genes and proteins which both deposit and then detect M6A. This is consistent with our modification results, where we see that the cerebellum has the highest levels of M6A and the highest proportion of modified genes and isoforms. And so this shows that even like with direct RNA sequencing, we can detect these different data types, but we can actually see that they're also consistent with each other as well. We found that the TUG1 long non-coding RNA had the most M6A modification sites in our human brain data. So TUG1 is a link RNA with many roles across various tissues, including in neuronal cell proliferation, apoptosis, and it's also a current therapeutic target for glioma treatment. Shown in red down the bottom here are the M6A sites along all the TUG1 isoforms. We can see there's actually 37 unique modifications here, which is in quite a contrast to the typical number of about 2 or 3 we observed per gene or isoform. Okay, so just to summarize, we identified large numbers of differentially expressed isoforms between the brain regions. Approximately half of our unique isoforms expressed in the brain were modified with M6A, and the cerebellum showed the highest levels of M6A out of the 3 brain regions. We could observe specific modification of genes and isoforms in a single brain region, and we detected changes in the M6A modification rates between isoforms expressed in different brain regions. And finally, the TUG1 link RNA had the most M6A sites in our human brain data, and this is something we are hoping to follow up experimentally. Okay, I would just like to thank everyone in my lab, especially my supervisor, Dr. Mike Clark, who is here today, as well as Dr. Ricardo, who did all the wet lab side of this and is speaking next. So please feel free to ask any other questions as well. Thank you.